Hello there, peace be upon you. Welcome to Rational Religion, where we make sense of spirituality. In today's episode of Believe You Me, we're going to be talking about random mutation and asking whether it deserves its crown place in Darwinian evolution. So, last episode we talked about natural selection, and briefly, what did we come up with?
We basically asked the question, is natural selection sufficient as an explanation to explain the trajectory of evolution and to reduce the improbabilities related to the production of complex organisms?
Yeah, and we said, it's a culling process, it's not a creative process. So if you want to explain how things actually got here, you need a creative process.
And if you want to check out that video, look there.
Or there.
Or there. Anyway, somewhere.
Yeah.
And in this particular episode, we're going to be looking at what is posited as the creative mechanism of Darwinian evolution.
Okay, so random mutation. What is it?
So if you imagine that we all know that we've got genetic code, okay? Mm-hmm. And genetic code is comprised of a series of letters, and those letters are formulated into words, which are known as exons, and those exons are formulated into sentences with genes. Mm-hmm. And then in chapters, you have your chromosomes, and the book of your life is your genetic code.
Right.
So in a very simple but very actually analogous way, we can understand the genetic code, the genes we all possess, and which it largely determine our physical makeup and our structure and other aspects of our lives as well and our beings. We can understand them as words of a book.
Yeah.
Okay? And basically, random mutation is the idea that if you want to get from Harry Potter to Lord of the Rings...
Mm-hmm. Just change the cover, right?
Just change the cover. Dementors, ring wraiths, basically same thing anyway. Mm. But if you actually really want to change Harry Potter to Lord of the Rings, all you really need to do is, you could, for example, change some letters. That's called point mutation. Or you could just rearrange some words, a bit of shuffling, okay? That's called exon shuffling. Or you could introduce some random bits of text from other books or plays like Hamlet or, King Lear and other Shakespearean works. Mm-hmm. And that's called horizontal gene transfer.
Okay.
Or you could take whole pages and just flip them around, right? Just change their position. That's I don't know what that's called, but I'm sure there's a name for it. So the idea is that you make all these random changes and effectively you should somehow, one of these particular copies should be closer to Lord of the Rings than it is than the Harry Potter was.
Yeah. So in Darwinian evolution, the idea is that you have phenotypic change over time, which produces new species.
Yeah.
And we know that in order to produce new species from a previous species, you need to have a decent amount of actual phenotypic change, which means necessarily you have to have significant genetic change.
Yeah, so phenotypic change just means your outward body.
It means you're actually
what you are.
In reality.
Your traits, your physical traits.
Mm-hmm. So you need to have a decent amount of genetic change in order to produce that, right?
Yeah.
And the idea then is that through these mechanisms, which you spoke about, maybe other mechanisms, basically the core of it is undirected changes in the existing code. You will produce something that is functional and new and interesting.
Yeah.
Right? So it's kind of like saying, so this is a famous example which Stephen Mayer uses a lot over in Discovery Institute. He says it's like going to a Microsoft engineer and making random changes in the Microsoft code and expecting iOS to pop out.
Yeah.
Right?
Yeah.
It's the idea that you make random changes in a code, which is highly specified information, and you're going to get something else, which is an end product in itself. But of course, it's ridiculous. We know, even from the computer analogy, you start making random changes in any code, you're not going to produce a whole new product. If you make random changes in a book, you can get a really bad book.
No, you're going to get an unintelligible book.
Yeah, that's true. This is not going to make any kind of sense.
And the reason for that is important, because you have to realise that the reason those words make sense in that particular order and those letters arranged in that particular word make sense is because there are lots of ways by which you could organise those particular letters in ways that make no sense. Yeah. And they far outnumber the number of ways that you could rearrange those letters to make a word that makes sense. And similarly, a string of words which produce a sentence, there are way more ways that you can arrange those words to create a sentence that is gobbledygook than there are ways. There's often only one way, potentially, to structure a sentence, and certainly whole paragraphs and chapters and pages, there's only one way to make sense. So what I'm trying to get at is the fact that the number of options that you have for changing and producing intelligibility and some form of advancement is infinitely smaller. So it's almost inevitable that when you make random changes, in other words, changes which don't have foresight in terms of producing something which is going to be...
They're not intelligent.
They're not intelligent, of an importance, of a greater advantage, then you're going to necessarily degrade the information. You're not going to be adding to the information.
Okay, I mean, that's what we're saying in theory. So that's the logical point, and that's been noted by people, for the last 50 years. I mean, 50 years ago, they said at the Wistar conference, they said, well, based on probability, if you start making random changes in the genetic code, given the fact that the ways of arranging your genetic code in ways that make sense are far fewer than the ways that they could be completely gobbledygook, you're not going to produce anything new, and you're certainly not going to produce the gamut of life over biological history.
Yeah.
That's the theory, but actually the wonderful thing about science is that it helps to confirm theories and it helps to confirm or disprove theories.
Yeah.
So what we're going to present today is actually proof that what doesn't make sense in theory doesn't work in practice either.
Yeah, absolutely.
So why don't you tell us about one of the first major empirical tests of random mutation?
Yeah, so you can often test things in science in what's known as in vitro or in vivo, so in the lab or in real life. Mm-hmm. So the first major test in a way came from in vitro. What was the experiment? So it was done by Doug Axe and Anne Gager a couple of years ago, and what that showed is that they tried to ask the question, well, how many mutations would it require to get one protein which is very similar in structure to its second protein? How many changes would it require to get it from that protein to that protein and adopt a new function? Okay, right. So this isn't about a function that the protein already does and it just gets better at doing it. This is particularly about one protein developing a new function, because that is the fundamental.
I guess I was going to say that's the subunit, isn't it, of evolution. That's the minimum thing that you need.
Yeah,
because over evolutionary history there must be billions really that must have happened of that. At
trillions.
Least, of one protein at a genetic level changing and becoming a new protein, new function.
Yeah,
because you have lots of proteins together make new tissues, make new organ systems, etc., etc. So this is like theoretical minimum.
Yeah, and that's called the development of a protein fold in effect. That is a series of protein folds to create a new catalytic site, a new function to that particular protein. So they took two proteins which are as identical in their overall shape and structure as you could possibly find, and they looked at this question: how many random mutations does it take? How many mutations does it take to get from A to B? And they showed that it required 6 coordinated mutations. And what coordinated means is it means all of these 6 mutations have to happen in the same lineage. Mm-hmm. In other words, they have to happen with the previous mutation having been locked in, and then the next mutation 6 times. Mm-hmm. Now, 6 may not sound like a very big number, but it's actually huge. And they calculated that through random odds, if this was to happen through a random process rather than a scientist with a pipette and a test tube, choosing it, it would take more than, wait for it, 4 billion years.
Which is the age of the Earth.
Which is the age of the Earth, and
which is a problem for evolutionary theory, because if you read your textbooks carefully, you'll find that most evolution happened on Earth.
Yeah.
So that's quite a severe constraint in the theory.
As they say in scientific terms.
Laboratory. I mean, what do people say to Doug and Anne Gager?
So what they said to Doug Axe and Anne Gager is that, well, hold on a second, you're looking at modern proteins. You're not looking at proteins which were well in the past, and therefore, were the kind of proteins that these changes happen to. You're rather looking at proteins which are too locked in to their existing structures and functions. They're too finely tuned to their function, which is an absurd idea because of 2 things. One, every enzyme in every protein is locked into its function, because by definition, the very structure of the protein confers its function.
So it has to be.
So you can't have this loosey-goosey protein that's kind of like also really specific to its function. But it's also really malleable and adaptive to be able to
it has to have a
else.
degree of specificity. However that actually occurs, it has to have a degree of specificity in order to do its function. That's what differentiates one protein from another.
Yeah, absolutely. So that was one argument. And then the second reply to that really is that if you're going to say that all testable aspects of Darwinian evolution all happened in the past, and that no current experiment can be conducted to test the theory, then what you're in effect saying is that the principal claim of Darwinian evolution, that random mutation can produce biological complexity, is completely untestable.
Yeah.
So yes, there is lots of evidence for evolution having occurred on the planet. Yes, evolution has taken over 1 billion years. Yes, organisms do change over time. There's no doubt about that. We see that, in the fossil record, there's a wide plethora of organisms stretching out millions of years. Absolutely. But to be able to say this one became this one, became this one, without there actually being something in the actual fossil record that shows you that mechanism of change, and then you claim that it's random mutation, you've got to show some evidence for that.
Yeah.
You can't just weave a story to explain an observation when the story's underlying premise is completely disproven.
Well, it's absolutely absurd that you have a theory based on two tenets: natural selection, random mutation. Natural selection, as we showed, is a culling process, not a creative process. Therefore, it cannot explain the origin of any biological novelty on Earth whatsoever. It can only explain why it continued to exist and didn't die out before reproducing. Right. So, natural selection, no claim on creativity. Random mutation is apparently the thing which produces creativity, but you find that to produce the smallest possible change that you could conceive of requires more time than we've had on Earth.
Absolutely.
So that's just theory, though. Maybe that's what you want to come on to. Okay, so we've seen that Neo-Darwinism is based on these two tenets, natural selection and random mutation. And it's supposed to produce all the biological complexity that we can see on Earth. And it's supposed to explain why things look designed without actually being designed, as we talked about in our last episode. Now, we've shown that natural selection is only a culling process. It's not a creative process. It can lay no claim to producing any biological novelty in the world because it doesn't produce anything. It just says, well, this thing didn't die before reproducing, right? Or its parents didn't. That's why it passed on its genes. Then you say, okay, well, the creative mechanism of Neo-Darwinism is random mutation. But random mutation we've seen in the lab, to the best of our empirical knowledge, requires degrees of coordinated change at a genetic level that it just can't produce probabilistically. The kind of massive widespread change at the protein level and the tissue level and the organ level that you need to explain evolution.
Yeah. So, we've got the in vitro evidence which backs up our intuition that random mutation isn't going to cut the mustard. But the question really is, what actually happens in actual biology?
What is cutting the mustard?
Yeah, so in vivo, in actual organisms, do we have any evidence from that? Maybe you can tell us about that.
Yeah, so really interesting experiments happening over the last 3 decades or so by Richard Lenski. So he basically has been running, him and his team, I guess, something called long-term evolution experiments, where to their credit, they said, you know what, we can do stuff in the lab all day long and we can theorize about things. But what we actually need to do is we need to actually see things in real life. Does evolution happen before our eyes? So they got vats of E. coli, it's a simple bacterium, often experimented on, and they basically just got them to reproduce in limited resources against each other in these vats over 3 decades, as I say or so. And they've actually produced about 70,000 generations, roughly, of E. coli reproducing. Now, let's think about the significance of this. What you're supposed to be seeing in Neo-Darwinism is that you have random mutation, natural selection producing widespread change. Apparently the story is that through undirected changes, we went from simple bacterium to you and me, speaking here now, through undirected changes and environments changing. Pretty tall claim when you think about it, but if it's to be a real theory which is confirmed, you need to actually see the evidence in real life.
In real time.
In real time.
Not in retrospect.
You need to see some evidence which you can extrapolate over a longer period of time. You should see something. So how much is 70,000 generations? Well, one human family reproducing over 70,000 generations gives you roughly 1 million years. Now, apparently there are about 6 million years between the supposed divergence from our chimp-like ancestors to modern human beings. Okay, so 6 million years for the overall thing. 70,000 generations, one family, is 1 million years. Of course, there wasn't just one family. Overall, they think there's roughly a community of 10,000 individuals who were part of that divergence. So that means, if you do the maths, well, roughly, if there are about 10,000 bacteria that are being reproduced over 70,000 generations, then that's the equivalent of what humans had over 1 million years of evolution, and that's a sixth of the total evolution you need to get, apparently, from a chimp-like ancestor to modern human beings. Of course, in reality, there are many more than 10,000 bacteria being reproduced in Lenski's trials, but we'll limit it to that at the moment. So what needs to happen in this time? Now, if you think about it, to get from primates to humans, you need to get much bigger brain, developed frontal cortex. You need to have altered birth canal in order to deal with a bigger brain. You need to be not on your knuckles. You need to be upright gait. You need to have all kinds of, less furry. Autistic sensibilities, spiritual capacities, all these things, even language, they say, came from just random changes. So there's a huge amount of stuff that happened in about 6 million years. So you can say, well, maybe a sixth of that happened in 1 million years. And therefore, as a rough gauge, we can think, well, there must be some kind of roughly ballpark equivalent change that we should be seeing in E. coli at the basic level when you're reproducing these things in vats.
So what do you see?
You see E. coli getting a bit bigger and a bit faster.
At reproducing.
And the most dramatic thing that they saw was that, presumably one of their flasks, they saw a little bit of fizz and steam coming off, if you will, and they found that one line was able to metabolize citrate, which is something that was in the solution that they had, and that produced a bit of a change.
Okay, so that's quite interesting. So they produced a new enzyme, citrase, which could metabolize citrate, because that would go against a lot of what...
Well, that's what people hoped. So they had these changes of going a little bit faster, a little bit quicker, probably hoping something more dramatic would happen, but they had to deal with what they had. And then the citrate thing, right? So they were hoping that these changes were happening because at a genetic level, you were getting genetic changes.
You were getting a new citrate metabolizing enzyme.
Yeah, or even that they were somehow managing to get more food on board, at that level, or they're changing themselves so that they can reproduce faster.
Yeah.
And they were hoping for some kind of real creativity at the genetic level, which they could then extrapolate over evolutionary history.
Yeah.
What they actually found is that what these E. coli were doing were breaking things. They were breaking down their own genome, and they were getting rid of stuff they don't need. They were just jettisoning them out. So, for instance, you can think of it like this. If you're driving a car and you need to go faster, I think this is an example given by Michael Behe. He said if you need to go faster, you could engineer things to give you nitrous, like in those PlayStation games, or a fast engine. I don't know much about cars, I'm not going to lie, but there are various technical things that you could do to make a car go faster, right?
Yeah.
Pedal down further is as far as I've got, to be honest.
Right.
So you could make new things. Or what you could do is you could make the car lighter by getting rid of, seats and passengers and all kinds of things to make stuff go faster.
That'll make you go faster as well.
Yeah.
But
and then when the police come after you, you go even faster.
But that and that involves actually the breaking of existing structures.
So you've got a framework which you're breaking down in order for optimum reproduction time in this.
So I think I read that, for example, they were getting rid of their flagellum because the flasks were being swirled, and therefore they didn't need their flagellum to move around.
Yeah.
So they just got rid of their flagellum DNA.
Yeah. So basically, whatever they didn't need, they got rid of.
Yeah.
So that's a real problem because they're seeing changes, but actually the change that they're seeing on the genetic level is the destruction of genes
rather than the innovation of new
rather
genes.
than innovation. And if you want to get properly new proteins and organ systems and evolution really going, you need biological innovation.
Yeah, I mean, we're not degenerate bacteria.
Yeah.
Put it simply.
Yeah.
Okay.
So you need a creative mechanism that actually gets to that point at the genetic level.
Absolutely.
Now the citrate thing. The citrate thing was quite interesting. So what they found, they're hoping again, I guess, that they would actually find that it engineered some new proteins that were able to metabolize citrate. What they actually found is that at a genetic level, these E. coli already had the capacity to metabolize citrate. But the difference is that they had a region of their genomes that was stopping them doing that in the presence of oxygen. So they already could do it, but when you have oxygen there, they had another gene which was stopping them, or another.
So they had a repressor gene in the
basically that was stopping them to doing that. And what happened was essentially that was moved. So you had a random chain that was moved. It is the sum total of the effect, and therefore that effect was lifted, and therefore they were able to metabolize citrate in the presence of oxygen.
Right. So it was the use of something they already had.
Yeah. Well, actually, it was the breakdown of something which happened to have a fortunate effect in that scenario.
Yeah.
So again, you've got 70,000.
Well, not even a fortunate. It was just a change. Actually, it conferred no survival advantage whatsoever.
Well, I mean, maybe it did a little bit, but it didn't. It's not going to get you new species.
No.
Yeah. So that's the headline news. The headline news is that the best that you can do in the classic Darwinian experiment, where you have same things in limited resources, just reproducing. They break things down in order to reproduce faster or get a bit bigger. They make minor variations in their organisms without making massive innovation.
So whether we look at it from the perspective of in vitro evidence or in vivo evidence, we have plenty of evidence that shows the underlying mechanism of evolution, which is, random mutation, natural selection, doesn't produce the complexity we expect to see in the world. What's really interesting is that the results of Doug Axe and Anne Gauger of more than 4 billion years in terms of the biological requiring one protein mutation and one new function for a protein, that's not surprising to people who work in the field. That came off the back of other papers also that had showed a kind of similar time frame of similar question. And so let's say, for example, the Doug Axe and Anne Gauger were 99% wrong. That actually it wasn't more than 4 billion years. It was only 40 million years. That requires one protein change from one protein to another protein, right? That is still prohibitively far too long for one protein change to another protein change to explain the biological complexity of life. And in terms of looking at it from a medical perspective, there's also an argument there to be said as well.
Right.
So, what would you say in terms of we're doctors? We treat people with HIV often. We treat people with cancers often.
Yeah.
And what are the kind of treatments that are coming out, and what are they premised on?
Yeah. So it's HIV is a good example that you've mentioned. So the treatment for that, the standard treatment, is called HAART, so highly active antiretroviral therapy. And the idea in HAART is that you give 3 different drugs which work in different ways to target the HIV virus. HIV virus, you may know, is something highly mutable, mutating unbelievably fast pace.
Yeah, viruses are like that. That's why we take a flu jab every year new.
Yeah. So what you have is you have an extremely mutable virus, which when you give just one treatment for, often, or it's capable of basically mutating in a different way so that treatment's ineffective.
Yeah.
But what they found is that if you give 3 at the same time, basically, even if they make mutations for one of the drugs, they won't make mutations for the other 2. Or even if they make mutations for 2 of them, just by chance, they're not going to make it for 3. So even on a medical perspective, we make this as one of our grounded assumptions that things don't have that high mutation rate in order to produce beneficial changes. That they can just do this, endlessly.
Yeah.
That there's a limit to what random variation can produce, and this is why we think, and this is what we use 3 drugs using at the same time.
Yeah.
And you said there's something
similar in cancer now.
Yeah.
They're using some of the immunotherapies with the same idea that cancer cells are also highly mutatable, and they very quickly can develop mutations against the drugs we're using. So now we're using drugs with multiple drugs with multiple lines of attack on the assumption that they won't be able to produce 3 coordinated mutations. So we're actually, if you think about it, we're actually saving people's lives on the premise that Darwinian evolution actually doesn't make any sense.
And a lot of people, though, they quote antibiotic resistance as being, how can you not believe in Darwinian evolution? Don't you know about antibiotics? So what would you say about that?
Well, a few things. First of all, complex organisms, more complex than bacteria, have a lot longer generation times than bacteria, first of all. And the second thing I'd say is also that bacteria in and of themselves, they undergo random mutation, and then they, you do get selection of a particular type of resistant organism. But the interesting thing is, very often, the resistant organisms are already there in the population. In fact, they found resistant organisms resistant to penicillin found in ancient Egyptian, sarcophaguses. And what that shows you is that, again, it's not the invention of a new mutation in response to selection pressure. It's actually the selection out of an existing thing.
I mean, even if it was, though, even if somehow organisms are able to adapt to stressors in this limited way, we don't see bacteria evolving into different bacteria.
Yeah.
I mean, over the last 70 years that we've had antibiotics, or slightly more, we haven't seen once something speciate.
Yeah, that's absolutely
under
right.
massive pressure of environmental pressure of antibiotics being used in a medical perspective. You don't see speciation. Now, the whole point of Neo-Darwinian evolution is that you have mutation and selection in order to produce speciation. The whole idea is it's supposed to explain how you get all these different species all across the world, which are so beautifully and delicately adapted to their environment.
Yeah, it's called On the Origin of Species, not called On the Origin
of trivial change within organisms.
Yeah.
So actually, antibiotics and bacteria cannot be used. Actually, they're a proof against, intuitively, the idea that you could have speciation just coming out of these undirected pressures and undirected mutations.
Yeah. In our next episode, we're going to discuss the third way of evolution, which is claimed by some biologists to be an explanation for the faults and the pitfalls that Darwinian evolution offers. But we're going to show that actually it's just Neo-Darwinism in a cheap tuxedo.
Peace be upon you.
Peace be on you.